The goal of this video is to describe a method that increases throughput while balancing effort and accuracy for the extraction of lipids from cell membranes of microorganisms for the use in characterizing both total lipids and the relative abundance of indicator lipids to determine soil microbial community structure in studies with many samples. In the field, you should account for soil heterogeneity, collecting soil samples in a way that will represent your site. To preserve the microbial community at the time of sampling, samples should be transported from the field on ice. Once back in the lab, remove roots and stones and break up clods by a coarse sieving. This also serves to homogenize your sample. Prepare for freeze drying by putting subsamples in appropriate containers and freeze dry as soon as possible. Once soils are freeze-dried, stored in a sealed container with desiccant until extraction. It is best to store freeze-dried soils at -80 degrees C. As a prep for extraction, remove freeze-dried soils from storage and grind. Methods for grinding include ball mill, ball meter, or mortar and pestle. After grinding soil, again, homogenize your soil samples thoroughly and store in the freezer. All labware must be scrupulously clean. Any residual detergent, grease, or dirt can impact the results by appearing as a peak in the final GC chromatogram. The extractions are carried out in 30 milliliter Teflon centrifuge tubes, which must be solvent rinsed. Add 2 to 3 milliliters of hexane to tubes and vortex for a few seconds. Decant the hexane to another tube and vortex.